Pleasure to welcome Professor Martin Gosling, who is going to round up the industry session this afternoon. So, Martin is CSO and co-founder of Enterprise Therapeutics. He was previously exec director in the respiratory area at Novartis, and he also graduated in I consider to be the best university in the world, Leeds. Give him that plug. We did dovetail there for a year. I can't remember meeting him. Probably met you in the old bar one night. Yeah, so please, Martin, your presentation title is "Fixing Broken Chloride Channels: Learnings from the Development of Treatments for Cystic Fibrosis." Thanks,
you. And thanks to the organiser for the invitation, particularly as I'm not a neuroscientist. I don't work on CLC channels. And I'm much more low molecular weight than gene therapy. However, I have spent the last 20 years working in the cystic fibrosis field, and I was asked to share maybe some of the learnings from the development of modulators of CFTR, and whether or not they could have any reference or influence on what you guys are doing. Looking at the talks that I've seen, interestingly, over the last 2 days, I think lots of the areas that I'm going to highlight you already seem to have considered. So I hope this is still of interest. What I'm going to do is briefly just give you, I'm sure a lot of you are familiar with CF, a little bit of an overview, really talk a little bit about how the basic science has kind of the understanding and driven drug discovery, a few of the things that I've kind of learned philosophically along the way, and then just a minor point about gene therapy in CF. So this is the kind of timeline. CF is the most common lethal genetic disease of Caucasians, an incidence of 1 in 2,500 live births. 1 in 20 of us are a carrier of the mutation. It's not a new disease. The first references to it were in the 18th century. There's a Northern European folklore that basically says, "Woe to the child who has a salty brow when kissed, for he is cursed and soon must die." And this is in relevance to actually what came out to be a biomarker for cystic fibrosis, which is the high levels of sweat chloride. In those days, a child would be lucky to see their 3rd birthday. The gut phenotype, this gut blockage, would be their demise. It wasn't until the mid-1950s that phenotype started to be unravelled, and the introduction of dietary supplements meant that these children started to live longer. And it was then the respiratory, the lung pathology, this inflammation due to mucus obstruction, failure of the innate host defense of the lung, clear infection that led to these cycles of infection and inflammation. The application of aggressive antibiotics started to improve that situation, so children now started to make it almost towards their 2nd decade of life. Probably the pivotal moment with the identification of the gene in 1989 by Jack Reardon in Francis Collins' lab, Malachi Su, of the cystic fibrosis transmembrane conductance regulator. For obvious reasons, it's abbreviated, it's the right mouthful, the CFTR. And shortly after that was shown that it encoded a cyclic regulated chloride channel. And that was kind of the beginning of the start. The next challenge was understand what that did in terms of regulating particularly this airway phenotype. I'll talk a little bit about that. And those kind of pivotal scientific milestones led eventually to the development of CFTR modulators, driven predominantly by the Cystic Fibrosis Foundation in the US, who put out a call for drug discovery efforts, which was answered by a small West Coast biotech called Aurora Biosciences, who was subsequently consumed by Vertex, that started the activities that eventually led to the registration of 4 medicines. So in terms of understanding what CFTR does in the airway, your innate host defense, you've got this mucus clearance that's continually going on. Mine's probably going a little bit faster than yours now, but it's there to protect you from all the inhaled pathogens that come with the 16,000 litres of air that you breathe. Mucus is stored desiccated in granules in specialised goblet cells. When it's released, there needs to be an amount of fluid such that it becomes a low viscosity water-like gel. It's actually 97% water, 3% solids. That water comes from the flow of ions. It's controlled by the coordinated activity of CFTR secreting chloride. Water's osmotically obligated to follow. And ENaC, the epithelial sodium channel, is kind of doing the opposite. Steam regulation, control of water. So you have this nice fluid gel, catches all particles. Obviously, when this channel is mutated, you lose that secretory pathway, you have this depletion of the airway surface liquid, the mucus becomes thick, static, you get mucus plugs, You can see on the bottom left, that's a mucus plug that has got a bacterial infiltrate in it, and there are a load of neutrophils, and they're trying to clear it out, but they just cannot do. So you get these cycles of inflammation that eventually lead to lung destruction. And unless the lungs are transplanted, the demise of the patient.
Can you speak a little louder, please?
Certainly. The channel has 1,480 amino acids, but it's got more than 2,000 mutations. Pretty impressive. Something's definitely gone wrong there. In terms of the mutations, they are characterized by their biophysical consequence into 6 pots, which you see here, imaginatively classed 1 to 6. Class 1 are mutations where you get no protein, so you've got kind of a stop codon. Class 2, you don't get trafficking. Class 3, you do get trafficking to the cell surface, but the channel refuses to open. And then 4, 5, and 6 are also mutations where you either get protein that's less stable, or so it's rapidly recovered, or again, it doesn't particularly want to open. Fortunately, from a drug discovery perspective, one mutation dominates, that's delta F508, that deletes 3 amino acids in the sequence. And 90% of CF patients contain at least one 508 allele. And that 508 results in a channel that doesn't traffic, and if you can persuade it to get to the cell surface, it's not very good at opening either. So you can see the bottom here, this was the kind of simple equation adopted by the field to try and understand how to fix it. The amount of current in any single epithelial cell is the product of how many channels you've got. Its open probability, i.e. how long it spends open, times the single channel conductance. The molecules that enhance the open probability in the CF world are termed potentiators. Those that increase the number of channels are termed correctors. There are a few people that will refer to potentiators as gating correctors, but that just confuses the rest of us correctors. So they're the kind of terms that one of the key questions in the field was, okay, we're going to try and fix CFTR using small molecules. How much do you need to fix? And actually, it's the patients that provide us some insight themselves. So here you have a graph. On the bottom is the percentage of wild type channel function, and on the y-axis, you've got the lung disease severity. Obviously, in a healthy individual, 100% wild type CFTR function, no lung pathology. The most severe mutations, such as 508del, which obviously you get very, very little CFTR function, it's probably between 1 and 3%. And the same here, I mentioned G551D, that's a class 3 mutation, but it stubbornly refuses to open. You have a very, very severe lung phenotype. 1 in 20 of us are a carrier. Again, we have no phenotype, despite only having 50% CFTR function. But there is actually a spectrum of mutations somewhere in between. Probably the most interesting are a set of male patients who tended to show up at sterility clinics because they had congenital bilateral absence of the vas deferens, yet they had no CF phenotype. When those mutations were characterized, they have somewhere between 10 and 25% CFTR function, which kind of gave the field a ballpark to aim at. You do not need to restore as much as 50%, somewhere in that kind of area is likely to give clinical benefit with respect to Vertex introduced the first molecule, ivacaftor. This is a potentiator in 2012 to treat a relatively small subset of patients. These are those class 3 mutations, so these are channels that have made it to the cell surface but not linked to the opening. They then followed that up about 4 years later with a combination of a corrector, so something that increased the amount of channel at the cell surface, with a potentiator. The first combination wasn't great, or can be. The second one was slightly better, Symdeko. And this then started to treat a much larger percentage of the population, around 50% of patients now have viable therapy. And that was followed up in 2019 by Trikafta, which, as the tri suggests, it's got 3 low molecular weight compounds, 2 correctors, and a potentiator. This then treats those patients that have a single delta F508 allele. So you can see here we needed 3 drugs to get effective levels of CFTR across an effective number of patients. There's still somewhere between 10 to 12% of patients that don't have a viable therapy with respect to CFTR yet. Perhaps interestingly from the Vertex experience and following up on something that Stuart said, the discovery program did not use an in vivo model, and the reason being there wasn't an in vivo model at that time. And when it comes to CFTR, mice definitely not. They have no lung phenotype, and I'm talking about knockouts, humanized mice, transgenics, every kind of thing that you can think of to try and generate one, other than the exception of the ENaC overexpressing mouse. So here, overexpressing that absorptive pathway does give you a CF phenotype, but knocking out CFTR doesn't. And the reason for that is different physiology. That airway surface liquid is controlled by different channels in mice to man, so the physiology doesn't stack up. There are now some larger species. They're not yet being used for active drug discovery. Ferrets are relatively new. Personally, from the people that work with them, understand they're quite aggressive. So I'm potentially not thinking about your animal house. The pigs that are used, these are not little pigs. These are 80-kilogram hogs.
But we could feed them the knockout mice.
Potentially. However, what Vertex did use to drive their discovery was near primary cells from patients. So these are human bronchial epithelial cells that are harvested from CF patients when they've undergone lung transplant. They're a relatively rare but precious resource. What you can do is you can expand them slightly in culture. They'll only go through a certain number of doublings before they'll totally de-differentiate. You then grow them on a filter. It takes about 3 weeks. You can see here it's that after a week you put them at air-liquid interface and they will fully recapitulate the airway architecture in terms of they have mucus-secreting cells, they have ciliated cells, all the kind of things that you would expect to see in vivo. What you can then do is cruelly sandwich them between 2 perspex blocks, and using the Ussing's chamber technique, we have some electrode, you can measure ion transport from one side of the cell to the other. And this is very much the benchmark technique of studying airway electrophysiology. And Vertex used this quite successfully to develop their molecules, and even have gone on as far to show that the correlation of what you can see in these assays very, very early on predicts what you're likely to see in the clinic. And here you can see that kind of data follow through for Kalydeco. On the left-hand side is the electrophysiology, showing that Kalydeco, or BX770, with its research name, effectively corrects the ion transport properties of those cells. In the middle, this is a measurement of airway surface liquid height. So these are cultures where you stain the airway surface liquid using Texas red, and then image it on a confocal so you actually could see more fluid. The third panel shows an assessment of mucociliary clearance in patients during one of the clinical studies. To do this, you get a patient to inhale a sulfur technetium colloid, which sits on the mucus, and using a gamma camera, you can measure its clearance. So you can see the acceleration of mucociliary clearance, and that then translated into significant improvement in clinical endpoints in terms of lung function and a reduction in the amount of infections that these patients experience. These assays are arguably been incredibly powerful to the extent that off the back of the safety database that Vertex have, they've been able to persuade the FDA to expand the licensing of Kalydeco into rare populations. Obviously, with 2,000 mutations, there are a lot of patients that are ultra rare. And Vertex have even been able to use recombinant systems, show that Kalydeco works in those systems, and they're able to go straight into treating patients. Organoids, also a really interesting area, particularly rectal organoids from CF patients, because one, you can expand them almost infinitely in vitro, and secondly, obviously, you can resample, same with nasal organoids. And again, not wishing to get involved in a pricing discussion, but Vertex medicines are incredibly expensive, and some countries are starting to insist or consider using organoids to check that the drugs actually will work via reimbursement for patients. Just want to go back to the low volume hypothesis, this understanding of what CFTR did in the airways and how that's actually helped with other opportunities to treat CF. So obviously, I mentioned there are other channels, so ENaC is the absorptive pathway. So other than just fixing CFTR, one other approach is potentially just block the absorptive pathway. So ENaC blockers are another way to consider in terms of increasing airway surface liquid and improving mucociliary clearance. There are actually even some genetics to support that premise. There are patients or individuals that have this PHA type 1. This is genetic loss of function in the ENaC. They have about 3 to 4 times the rate of mucociliary clearance normal individuals. The images you can see here are under a bronchoscope. They have so much airway surface liquid, their airways actually glisten. So there's some genetics to suggest that blocking ENaC will actually improve airway surface liquid height and mucociliary clearance. Unfortunately, from a pharmacological perspective, we haven't quite got the drugs right to do that yet. Interestingly, there's also another chloride channel expressed in the airway. So this is called a bypass or surrogate approach. This is a calcium-activated chloride channel as opposed to a cyclic AMP-regulated one called TMEM16A. So here, the strategy would be to enhance chloride flux through TMEM16A as opposed to CFTR, to provide that same opportunity to get more fluid into the airway. This is actually some work from my own company that developed first-in-class TMEM potentiators. The reason for the sheep is this is actually mucociliary clearance measured in sheep, not a patient. So this is a healthy sheep that was administered the molecule ETX001, and 4 hours later, sulfur technetium colloid was puffed down into the sheep's lungs, and then image clearing. And hopefully what you can see there is that the ETX dose-dependently increases mucociliary clearance. And this is now in clinical trials with Genentech. One final point I want to make as a pharmacologist, my primary training at Leeds, is about having a good toolbox. So you can go with TMEM16A. These are 6 molecules that were available in the literature, that the majority of the literature have used to suggest or support a role for TMEM in a variety of processes. EACT is an activator, the rest are reported to be inhibitors. This is our work using an automated electrophysiology system, just characterizing what these molecules actually do to TMEM16A. And what you can see is this bit of a fruit salad in terms of the activator not activating. It activates 2 other channels, but certainly doesn't activate TMEM16A. Some of these inhibitors inhibit other channels, and actually only Ani9 looks to be reasonable. So I would encourage you to, one, really characterize your molecules. If you start to look even further, this is some profiling we did on GPCR, so you can see that again, these molecules are far from selective. So if you do start to look for pharmacological agents for CFCs, then do make sure that they do what they should on it. So finally, just a couple of points about some of my learnings from working in CF. It's taken time to get from the identification of genes to therapy. I'm sure things can go faster, but they do also need to be done properly. The basic science and understanding of what the channel does has really driven the drug discovery efforts. The ability of academia, pharma, biotechs to work together has really helped. And I cannot underestimate the contribution that charitable foundations, particularly Cystic Fibrosis Foundation, have made in terms of CF, in terms of maintaining the profile, seeding the drug discovery, in some cases actually paying for it. Obviously, my view of in vivo models here is not great, based on the information that I shared. In this case with CF, they're useful, and they might continue to be useful in terms of understanding the pathogenesis of the disease. But I put not critical. If you've got human reagents, then here's an example. And I think Stuart mentioned where you can just go straight to ask the question. In terms of gene therapy or gene editing, I'm sure this is the future for treatment of CF, but CF's already over 20 years into gene therapy, and arguably it's a monogenic disease that you can actually access topically, i.e. you can inhale products. It's yet to be successful. And the major reason for that seems to be that you're trying to get gene products commonly using things like viruses into cells that have evolved for millions of years to keep those things out. So I'm sure it will actually be successful in the long term. But at the moment, it's still struggling a little with that primary question of can we get it in? With that, I'll stop and just thank the